right now. And I want to do on the Learn Where You Live series, you'll each be given a slide deck, and when we're in breakout rooms, that'll be explained in more detail. But I wanted you to know right up front that this first page, this is a link to each session. So if I click on that real quick, I'll show you if I do it right, or maybe not. Well, you know, I'm just lying to you. That's the way it's going. It will take you to the advertisement doc that underneath has the list of what is being presented for each day of the week. And it has the link to the Zoom meeting on it right there too.
If you hit present up in your top right, the link will go hot.
Oh, is that what it takes?
Yeah.
See? There you go. Thank you very much, Jim. So this is what is on your slide deck so that you always have access to that. Okay, so the next thing is on your, where your name shows up. And I don't know if you're familiar with Zoom already, this is old news, it's not new news. If you make sure you click on participants, and then you'll see your name. And if you right click on your name, if you can hover over your name and click rename, we're going into groups, or we will go into breakout rooms, and they're divided in K4, 5, 8, or high school. And if you'll put your preference on there, there's a possibility you won't get your preference only if one is too overloaded. Does that make sense? If one group has too many, I'll try to get a balance, but and I have everybody's registration, so I know what grade you teach. But if you could rename yourself, that's very helpful. And then basic information is please remain muted unless you want to speak, just in case you have background noise, because I have a dog, so I'm very familiar with how that works. And we love to see your face, but you're not required to show your face while you're here. As far as attendance and everything goes, that's going to be recorded on the slide deck. And then if you have any questions throughout any of this, please go ahead and post them in the chat. One other piece that I wanted to show you before we introduce everybody is your participant folders. I think everyone should have received a copy of your participant folders. I'll do this again. Hopefully it won't be as messy as last time. So everyone has their own folder with just your first name and your last initial. And I will show you mine. So within your folder is appendixes E, F, and G, which give you the progressions on the DCIs, the science and engineering practices, and the cross-cutting concepts. It also has in it a comparison document where Montana state standards align with Next Gen, or if they don't, and if they have Indian Ed for All added into them. And then this component right here is your slide deck. And I'm going to open that real quick. You'll get more details on this later. So for today, the directions are all on this second slide. But for today, this first one, September 15th, what is watershed? You'll put your name right here, and this will count as your attendance. And this is how I'm going to record OPI renewal units. So does anyone have any questions on those components so far? No? Okay. So I'm going to do a quick poll. And please wave me down or something if you don't receive it. So there's two parts of the poll. The first one is if it's not your grade component, you just mark this isn't my grade, and then go to the one, and then kind of just put how familiar you are with the 3 dimensions and Next Gen. It looks like we still have a couple people in there. I think everyone's in. So I'll share results. Can everyone see those results? Awesome. Thank you, Katie. Okay, so We've got a mix, and that helps our presenters a lot in order for them to determine how they guide the breakout rooms. The last thing I want to do is introduce to you the people that are presenting, the teacher leaders. So we have 3 teacher leaders that will be your group leaders. It's Christine O'Shea, and this is wrong. This will be K4. Sorry about that. And then Chris Pavlovich will be 5th through 8th grade, and Bill Stockton will be hosting the high school group. Okay, Chris Pav, you want to go ahead and take over?
Do. Hi, everybody. There are a lot of familiar faces, which is really cool. So I just want to give a wave because we get to see everybody's whole face, even though it's a little box, it's not half the face. I enjoy that. I'm going to introduce Raylan Brandel. I'm lucky enough to call her a friend, and Montana is lucky enough to have her as a professional in the state. She's the director of the Clark Fork Watershed Education Project, and she's here to talk about her work in Montana with watershed education and in her scientific endeavors as well, exploring watersheds, collecting data, and monitoring watershed health. And so we owe her a lot in Montana for watershed education. Welcome, Raylan.
Hi, everybody. Thank you, Chris. Well, I'm very grateful to be here today, and it is really fun to see such a diverse group. I was looking at the list of participants from around the state, and it's nice because as the Clark Fork Watershed Education Program name implies, we tend to work in this watershed a lot. So we have branched out over the years to be across the state of Montana. So now today with this virtual meeting format, it's kind of cool. I think we've hit most of the watersheds of the state at this point. So pretty neat. So I guess just a little bit about me and about CFWEP. I call myself, I have a foot in each world. I have a science degree and I have an education degree. And my master's work has really been an interdisciplinary master's. So I've done that blend again, where I research my students and the impacts that our curriculum has with our students. And then I also do a fair amount of research on what I like to call the fun science or the hard science out in the field, in which we do various macroinvertebrate counts. We've been looking at beaver mimicry devices and the use of those in high mountain stream systems in order to maintain water in the high mountains. You guys will have some work with that later on in this series. From not me, from other scientists that are working on it. And then doing a hell of a lot of mentoring with college kids and high school students, particularly in a phage discovery project that Linda Rost is also a member of. And so I kind of am a jack of all trades and an expert at nothing. So it's kind of fun to kick this off in the watershed sense, because as we were developing and designing this series, we thought, you know, what do we think that teachers would like to engage in? And so the title Know Where You Live came into our thinking, because we were thinking about everywhere we are in Montana, there's unique atmosphere, unique ecology, unique environment. And so we thought we want you to take the model that the Clark Fork Watershed Education Program has had and apply that into your particular area. And then when we were looking at it from an ecological sort of point of view, we thought, you know, a watershed is a great boundary to use in order to talk about knowing where you live. And so the materials that I've sent you that you have in your background materials are really a set of materials that we're using this year online with our students. So you have a series of videos and story maps that are from ArcGIS, so you could follow our model, and then you could adapt that to your own watershed. So looking at where your watershed boundaries are, where does your water come from, what are the particular issues in the history surrounding your watershed, and then how can you get your students out to engage in some of those questions. The scientists that actually follow me or follow in this series later on will be your content experts. We are more like the backbone and the big picture, and then the content experts are coming later. I do have with me my colleague from Missoula, Dalit Gusillo, and she does also a fair share of research in the summertime looking at osprey and using osprey as a method of monitoring metals in our watershed. And so she'll talk more in detail about that. I I'm trying to think of other things to say synopsis wise, Chris. I think you said you were going to facilitate us because there's too much for me to talk about.
Absolutely. Can you talk about any of your current work that you have?
So the biggest work that we just completed is we studied our students' responses to the watershed programming. And so we started to look at their attitude changes towards the environment and attitude changes towards science. And I'm happy to report that after 10 years of looking at this data, we did have a bump, that we had a medium effect, a Cohen's d medium effect in environmental attitudes, or changing towards a positive attitude towards the environment. And so that lends a lot of credibility to the work that all of you and the Sept program have been doing, for sure, is that, you know, when you get kids outside and you engage them in looking at their local area, they do start to care about it. The tenet that we use in Sept is we call it early, often, and always, meaning that we need to engage our students early. So I love that we have great representation of K-4 teachers. And then often, meaning that we need to keep doing it over and over. So even if they saw something about macroinvertebrates, say in 4th grade, I think it's totally appropriate to repeat that in 5th, 6th, 7th, 8th, 9th, 10th. And then always, meaning as we do our public outreach as well. And so that's work that we just completed. Delita and I will be looking at follow-up studies to see how long does the effect last, and then also how long does the change in their content knowledge last. Because as teachers, you know that we have that summer effect where they fall off and they don't remember anything that they learned in 5th grade when they get to 6th. So we've started to examine looking at our particular content related to the Clark Fork watershed, how much of that retains in their memory as they move into later grades. And do they remember that this, what happened in the Clark Fork watershed was really a large-scale restoration project, and that there was historically extreme mining damages, and that now we're cleaning those up, which of course is important to the people that are funding our work. Presently, today, my colleague has my biodiversity students out, and they are, you know, just doing a regular biodiversity 101 sort of biology class, or I guess it's 151 biology. And we do have 2 students that are working on an ecology project related to trying to get at invasive weed species that are attacking the restoration projects or the remedy projects in Butte. And so what they do with that project is we're specifically looking at cheatgrass, which is a very difficult grass to get out of our cover crops that we use to remediate our mine sites around Butte. And so cheatgrass will take over a site, and then it ends up adding to a lot of active erosion. And so those students right now are setting up transects to study how much is there, and then looking at various amendments to soil and different treatments in order to get that cheatgrass to go away. One of the problems that we have in Butte is originally a lot of the caps were planted with non-native grass species. And so those non-natives have made a space for the weeds like cheatgrass to come in. So that's a project that they'll be working on this semester, and they've got a nice greenhouse kind of set up that they'll be playing around with. So hopefully next summer we'll be out on the ground doing a few test plots. And then our phages project, we're in the process of cataloging and identifying all the phages that our teachers have gathered over the years. And that's a huge undertaking. And I am lucky to say I don't really have much to do with that other than to look at how the teachers are reacting to that project. They are enjoying it, they're hating us, they're excited, they're whatever, they're checked out. But right now it's kind of an interesting thing to watch from afar to see how that cultivation of the phage library, as we're calling it, is going.
Thank you, Raylan. So we're going to start with a couple of questions from trainers. And I encourage teachers, if you have questions about the content that she's talking about, how it relates to your area or her area, please put that in the chat box as well.
Hi, it's Christine here. Raylan, I'm just wondering, you answered a lot of the questions that I was going to ask. So I'm just wondering how long has CSWAP been in existence, and maybe the connection between what you're doing and the local community? Like, how do students engage with the local community as far as outreach goes?
We started with the pilot project in 2004, and then started officially with grant funds from the Natural Resource Damage Program in 2005. And so we are at our 15-year anniversary right now. And the way that we've originally started is, the idea was we would go to the schools of the Clark Fork watershed that were immediately affected by the Superfund cleanup. And so we touched all the schools in Butte, Anaconda, Deer Lodge, Drummond, Bonner, and then the Missoula schools. We also worked with the schools that are part of the Blackfoot watershed, or the Blackfoot River, that is, of course, a tributary to the Clark Fork. And so all of those little schools, we work with them as well. And that, how we do it is we traditionally have gone to the classroom. There's 4 days of in-class instruction and then a 5th day of a field trip. And this year, of course, everything's different. So we have a variety of things that are going on this year. The materials that I'm sending out to all of you, we have the story maps and the digital offerings that we're giving. We have some virtual field trips that we're going to host for East Middle School next week. So we'll see how that goes. I have no idea how that's going to be. We've shot a lot of videos to show students what it looks like in the field and how we do that work. And then some of the things that we try to do is also include programming for the summer, which Dalit will talk about some of that summer programming. And then we do a fair amount of service work. So we do a cleanup day. We call it Cleanup Blacktail Stream Day or Cubs Day. We do various stormwater outreach projects and a stormwater art project. So our students teach others about how important it is to keep track of stormwater and how to keep it clean, and the things that they can do just at their own homes for that project. It's never linear. I would say it's always moving, and we're constantly trying to evolve and engage people where they are. We've also built a whole bunch of different websites. The Science Partners website that I sent the link for you guys to was one that we built early on with our STEM faculty at Montana Tech, and some of you on this call were involved in that. And then one of the ones that we've just completed is called Pit Watch, and that one is about the Berkeley Pit, and it's 100% of content related to the Berkeley Pit. And for those of you that don't live in Butte, you probably have at least heard of the Berkeley Pit. It's one of the infamous Superfund sites of the country. And so we've been trying to talk about how that isn't this horrible toxic waste site. It's a managed site. It's, it could have been better early on in the decisions that were made, but the management of that site right now is as great as it can be. And then all the incredible science that is going on with it. And for high school chemistry people, I think it's a really cool and engaging topic, because the things that are happening in there right now with changes in pH and stuff is pretty fascinating.
Thank you so much, Raylan.
Hi, Raylan. I've got a quick question for you as well.
Sure.
So I know that you do a lot of work with macroinvertebrates and looking at data after you guys have some remediation. And so could you talk a little bit about the things you guys are seeing after you go in and remediate an area, and what types of macroinvertebrates you're looking for, and why that's a good indicator of stream health?
Sure. So I want to make the caveat that the CFWEP data, it's like a Polaroid snapshot. So our students are collecting that data, so we don't at all claim it to be scientifically pure or sound, but it is a great snapshot. We have worked with incredible associates over the years, and are actually part of some of their publications. But the snapshot data I was looking at today, I was looking at data sets from 2006 that our East Middle School students got, and the scores were abysmal. They were in the totally destroyed zone, if we were going to use that as the marker. And we were only catching things that were very pollution tolerant, like leeches and black flies and blood midges. So each of those organisms are, as I said, pollution tolerant, but they're tolerant to many kinds of pollution, one being low oxygen condition, and the second being high metals conditions. Lots of macroinvertebrates can't live when there's a lot of metals in the water, copper being one that's terrible for them. So compared to the data sets that we got last fall, where we were, the biotic index scores and the EPT scores were in the good to very good range. And so we were finding stoneflies, which I have to say, we didn't find a stonefly for 10 years of my experience on Silverbow Creek. And so having them back in the last 3 years is pretty incredible. Yeah, Chris is given, it's been pretty incredible. And now, for those of you that are familiar with the stonefly, it stays in the water for, there it is. Josh has got one. So it stays in the water for a long time. And so if you have a season where you wipe them out, they're not there to repopulate. So you have to have them there for more than 1 year in order for them to be there. So now we're starting to see them, and they're bigger. So they're getting to their later instar stage where they will hatch out and be adults and be able to repopulate the stream. So, and then, like I said, that score was much higher, and we're getting more sensitive mayflies and also more sensitive caddisflies. And then we still have the leeches and the blood midges and everything, but they're not the only things represented in the sample. Macroinvertebrates are a great way to monitor stream quality because they're easy to catch. It's fairly easy to do the identification, and then they are always in the stream. And if they're not in the stream, it's generally because there's a major problem.
Thank you.
Raylan, thank you so much for sharing. That's just a slice of all the work that you do, and it's so rich and there's so much to it. It definitely shows what we can look at as a lens, as watershed as a lens in our classrooms at any level. And so that's what the series is about, is looking at watersheds and thinking about how we can teach and learn about our place and learn where we live. And so I really appreciate that. Would you be willing to introduce your colleague to us?
Yes, I would love to introduce the lovely Delete Cusio. She has been, honestly, she's my superhero. she takes on, she teaches in all of the Missoula County Public Elementary Schools, and then she also takes on Clinton and Bonner for us so that we don't have to travel quite as far. And I have to say, honestly, she has taken the Missoula area programming from kind of a fun, let's do a cookie mining activity to hard science in the 5th grade classroom. And she's been absolutely incredible. And also, like me, she has a foot in each world. Her background is in genetics, and I'm going to have her tell you about her science background more than me. But she's been a passionate educator for a long time, but also able to do the the osprey blood sampling in the summertime and engage with the researchers at U of M about that. So that's Delete.
Thanks, Raylene. Can everyone hear me? My background is, like Raylene said, is more from the sciences. I have organismal biology, conservation biology degrees, and got into education. Funny enough, my dad would say that it's from my genes because my last name is in Russian means teacher. So I will just take that. And so education is in my blood and science is in my heart. So it's a great combination to be working with CFWEP. So like Raylene mentioned, during the academic year, I work on campus teaching college students science education. What is science? Why is science education important? And why is it important not just for the few that want to become scientists? And then I take these guys with me to the classrooms in 5th grade, and we actually have stretched the program to a 2-week-long program in normal times, and build curriculum around it that fits with the Common Core, ELA, and math. And so the kids are really immersed in a 2 weeks program and the story of the Clark Fork watershed that ends up with them asking, well, is our watershed healthy? And they go into the field, collect their own data, and then compare it to standards to answer their own question. And funny enough, we also use data from Butte, from Raylene, from Butte pre-cleanup. So they're not only they compare it to standards, but they can compare it to what kids their age have done 15 years ago, you know. And so it's a good story. Anything that you teach as a storyline is clicks, right? The kids really enjoy it. I'm also lucky that then I can keep on doing what's in my heart, the science, during the summer. And I joined the crew of the Montana Osprey Project, where we go and we monitor ospreys in this watershed. And the idea is that ospreys are just a great sentinel species. They are, where there's water, they're ospreys, and they're the top of the food web. And so we can go and take blood samples from the chicks that were born in this watershed. We're fed fish that are within a mile diameter from the nest, so we know where the fish comes from, and look at what they have in their blood, and that is representative of what's in that water. This work has been started in 2006, so we have pretty good long-term data, trying to go back to the same nests if they're active, if they're successful. As you guys can imagine, there are years where it's not the greatest, and there's a lot of factors that we don't understand. But for the most part, we go to the same nest in the watershed, all the way from Warm Springs, close to Butte, and down to, at times, all the way down to Frenchtown. And we go up to the nests, pick up the chicks, take them down with a bucket truck, band them. So every time you put out your hand on a bird in the US, you actually have to band them. And then we take a blood sample and send it off to, well, we don't send it off, I actually take it by foot, because it happens here at the University of Montana, so it's fun. I walk over to the chemistry lab, bring them the blood samples and feathers and eggs, if we have eggs that haven't hatched, and they do the magic, and we get numbers. We look at the 5 heavy metals, zinc, copper, cadmium, lead, and mercury. We get trend data. Now, why is it important? In my perspective, is that one, osprey is one of this animal that is equitable in our state, and actually all over the world aside of Antarctica. Like I said, wherever you have fish, you have osprey. It doesn't matter if it's a good fish or a bed fish or healthy waters. If there's a fish in there, then you'll have an osprey catching it. And so anyone who picks up their head up and look at the sky, and here in Montana, it's during the spring and summer and early fall, you can see an osprey. You don't have to be in a remote area, beautiful scenery. You can see an osprey anywhere you are. And so it's an easy way to talk to anyone about their surrounding, right? Have you ever seen an osprey around you? You have. And then talk about the interaction with the ecosystem of that bird and the biomagnification, basically how the heavy metals get stuck in the fat tissues and the blood of the osprey or any living item, right? And it doesn't get excreted. And looking at it and talking about what's the implications for us, what's the implication for the ecosystem, and so on. So during the summer when we do that work, we always have, not always because this summer was different, we always until this summer had a component, an education component. So we have kids from camps coming and seeing our work as we do the work, and you can't get tired of coming down from bucket truck with a chick in your hand and having the kids see it for the first time. It's just this mesmerizing, like, and it doesn't matter how many times they see it. Actually, it doesn't matter because I have done it now for plenty of years and I still get excited. It's just, I don't know what it is. Maybe the puppy thing. I don't know what it is, but it's just exciting. And slowly in the past 3 years, I've realized also with the idea of equitability, there are a lot of kids that are not necessarily in camps, right? If you have more than 1 kid or 2 kids or 3 kids, sometimes where you're better off, the family is better off keeping the kids at home. And so we've started making these community events. So we post about it, and we have people coming from not only from Montana, but from different states to come and see us do the work. And so it's a really great outreach work. What we're working on right now that I'm super excited about is that we're working on a 7th grade curriculum, finally looking at our data. And so looking at NGS standards, realizing that evolutionary adaptations are taught in 7th grade. And osprey is just this amazing species to talk about adaptations to their natural environment. Then we're working on this curriculum where we'll look at an osprey as a perfect specimen for evolution adaptation, and then a component of the ecosystem interaction, like how the osprey interact with the ecosystem. And we'll have a pretty intense portion of data science, because the more I get exposed to data science, I realize this is really what we need our kids to learn, is to look at data and understand how to visualize it and how to understand that data, because everything around us is data. Everything, even they all have their phone, right? And that's data right there. And so we're working on this curriculum, hopefully having it done by April, May. Don't hold me accountable, Raylene, on this. I'm worried. And we'll actually give you guys raw data to work with on this platform that's called CodeApp. And if I have time, I don't know, Chris, Michelle, Raylene, if I have time, I'd be happy to share. It's very raw because we're still working on trying to understand the data itself. We don't have much data. If you think about it, you go onto a nest, we maybe have 20, 30 nests a year if we're lucky. We usually try to get blood from one chick, and that's enough for the funding component, but one chick is enough to tell us what's representative of that area. And even if you look at it pre-2006, you still don't have many data points. So it's really, seeing a trend is very difficult. But the idea would be, and I'm going to try to share my folder, my screen, wish me luck. This one. Can everyone see this? Yeah, perfect. And so this is an amazing platform where, if you can see right now, I have, and this is not the way the data is going to look like, but right now you can see that I have 132 cases over years here. I actually don't know if you guys can see my pointer. Can you see my pointer? Yeah, okay, great. The location of where the nest is, and then the average of these different heavy metals. So average copper and zinc and so on. And what you can do in this platform, which I've very much fallen in love with, is you can pull up a graph and it's in real time, and you can drag whatever you want. So the idea is that not just coming and asking questions to the kids, is having them just come and go and grab stuff and tell me what you see. And the idea here is that it's a low floor, high ceiling. Anything that they visualize and they verbalize it is a good answer. And so, for example, if I take year and I drop it down here, and I take, I don't know, copper, and I put it over here, and I'm going to put the copper in, numeric. And I'm also going to get, so you can have 3-dimensional, I'm going to take the locations and add it in here also. You can see, and so, right, the problem here is that we have way too many locations. So the idea is I want to clump these into general areas, upper Clark Fork, mid Clark Fork. But you could have the kids just go over and look at it. And right, like, what do you guys see here when I look over the year 2006 to 2019 copper? You can say anything you want, it's probably right? But oh, look at this, in 2013, the Tower Street, which is here in Missoula, is kind of an outlier. What happened over there? They can look at copper, and put zinc over here and look at that. It's pretty ugly, but the idea is that it's really interactive with whatever they want to do. And we'll, of course, have a lesson plan with eventually taking them and asking the right question. And the idea here is that our data is not perfect. This is not climate change data where we have millions of data points where you see a trend line. It's hard to not see it. But that potentially will allow conversation about what we have, what we don't have, why we don't have it, And that's where we would come back in either in the classroom or interactive via Zoom or emails of like question, and we could tell them how real science is happening. Why don't we have the answers for that? Why would we want to have the answer for that? And the data will be progressing, right? I have the blood samples in my refrigerator for 2020. That's going to add on into here. So that's in general what I kind of wanted to share with you guys. I don't know if there's any questions. I kind of dumped a lot on you guys.
We have some questions in the chat that Michelle's going to read through and ask. I want to give everybody a chance to type a question to either of our experts. Thank you so much. That was an amazing, I saw so many smiles and people leaning into the screen because they were so interested. And I mean, if we could do that on a Zoom meeting, get people to lean in at, 4:40 on a Tuesday, yes. This is what, science looks like in real time, in the real world. And that's what we're trying to connect students to in the classroom is science. And so thank you so much. Trainers, do you want to open up some questions, and we'll give people a chance to start typing away.
Hi, Christine here. Dalit, I was going to ask with the ospreys, what do you think the overall, or what do you hope the overall impact of your research is going to be? Like what can come, what do you hope is going to change because of that?
Yeah, that's actually a great question. So there's, when you think of science, there's the pure science where you just go and ask a question because it's interesting. It's just pure exploration. And human society has advanced tremendously with these kind of questions. For me, coming from conservation biology, having more of this drive of activism and trying to figure out how human impact their environment, this is a monitoring study. This is not a research study for research, although I have many questions I would love to ask for research, but the funding is challenging to say the least. For me, this is monitoring to look at trend, seeing if anything changes, and making sure that that information goes back to the communities, right? And so, for example, in Drummond, we've seen a lot of spike in mercury levels. And we have some insight of why we think that is the way it is. But more importantly, we wanted to make sure that the community over there knows about it so that they're informed, and informed citizens can make change, right? And while we don't know what mercury does to ospreys, high mercury in the blood of ospreys, we do know what it does to human brains. And so purely without having the data, if they have a lot of mercury in their blood, does it mean that we have a lot of mercury in our blood? I don't have that answer, but what I do know is that it's very high over there. Well, it's not high, it's almost undetectable in other places, and that is a cause of concern. And so these citizens can go back to their representative and say, we want this to be looked at, like what is going on with this. And so I think that what CFWEP is doing in education and monitoring in this watershed is about education. Like without data is knowledge, without knowledge you can't do anything to affect your society, your small community. And if we recall the fact that we are all talking about watershed, the main word of watershed is water, and unanimously we all can agree that without water, without clean water, we cannot survive. Selfishly, I'm not even going to talk about all these ecosystem services that we're potentially getting from the watershed, and we have no clue what they are. Selfishly, if we don't have clean water, even if it's the water you buy in a plastic bottle in your shelf, it's coming from nature, it's coming from your ecosystem, then we're goners. And so for me, it all goes back to education and getting people to understand that scientists are not here to come and get them. Scientists are not here to we don't have an agenda. My agenda is to have, a healthy, beautiful life, and for all of us. So it's kind of like, I think that's something we should potentially all agree on.
Thank you so much.
Hi, Dalit. I have a quick question for you as well. So you mentioned that you guys are going up and collecting this data, and that you've been involved the last few seasons collecting that data. When you yourself start looking at the metals, is there any trends that you're noticing just off the top of your head, or just the one instance in Drummond?
So I should start with the good news is that all of the heavy metals, but mercury, is really low or even undetectable in blood, in osprey blood. So that's the good news. The mercury is high at times and in places, especially around Drummond, and it still is. And we think that that's because of so mercury was not used for copper mining in Butte. It's used for gold mining, and there are a bunch of gold mining up in Flint Creek, active and non-active. And unfortunately, once you leave a mine, the heavy metals are still in there and still keep seeping into the system. But another interesting point, and I'm still not sure about it, it's all correct, and it might be correlations forever, but right now, another thing that's pretty cool that we saw is that it looks like that, was it last year? Like last year, they started remedy work above Grand Cores. And it looked like this. It looked like they were building a highway there. Like there was nothing natural about that area, and the blood, the heavy metal levels in the blood of the chicks at Grand Cores were extremely high, so they spiked. So that was really interesting for us to see. Unfortunately, this year that nest was not successful, so we don't have data for this nest, and who knows why they were not as successful. But we don't have data for this year.
Thank you.
Can I add a piece, Bill?
Yeah.
So Delita's talked about how the osprey, they're the top of the food chain. So another study that we were able to participate in for quite a long time was being done by the USGS, and the gal's name is Michelle Hornberger, and I will make sure to get a link to you guys so you can look at this study. But she was looking at a caddisfly called the Hydropsychidae caddis, and it's always an early colonizer of streams that have been disturbed, and it's pretty metals tolerant. Like it can tolerate copper, it can tolerate all this other stuff. And so she actually was looking at the heavy metals in the bodies of these caddis, and turns out they had the same trend. They were having, in the areas that were heavily damaged, had extreme amount of metals. And then now, as we've been fixing things and moving on, the metals that she's finding in the bodies of these caddis are now also decreasing. And so if you think about that, caddis is eaten by the fish, and the fish is eaten by the osprey. So you would expect to find those metals with the caddis flies. She wasn't looking at mercury, but she was looking at cadmium, arsenic, zinc, and lead. And zinc tended to be extremely high in the caddis flies. And then, like I said, we had students that actually she mentored for a few seasons, and they would take the caddis and take them out of the damaged area and put them in a clean area, and then sacrifice them to see if they could clear the metals from their body, and turned out that they could. So that was kind of a cool project, but it just adds to the piece of like, where is it coming from? Well, it's definitely food chain effect.
It also looks like we have one question too that maybe one of the two of you could answer. And the question is just looking at if you live in an area like the plains where you have low moving water and maybe not osprey nearby, what other species maybe that live in those types of ecosystems could, you could get students excited about kind of talking about this kind of similar type of material. Do you guys have any ideas?
So I would start by making sure, looking up and seeing if you don't have ospreys, because seriously, if I were to put a tub in my backyard with some fish that I caught in a puddle, I probably would have an osprey coming and checking it out. We have ospreys migrating and spending their winter in the nastiest ponds in Texas next to like chemical factories, because there's a fish in that lake, or many fish in that lake. So I do believe that ospreys are there. They might not be as common as next to a free flowing river, but I would argue, and I clearly like ospreys, but look up, maybe you do have an osprey over there. But I think you can, like, I think you can get excited with any kind of animals whether it is macros. And I know that kids really get like disgusted, but in an exciting way, when we do find a leech. And then you put the leech on your hand and they see that it doesn't eat you up, and you can move on with that. And so I think you can potentially look at the lower part of the food web, and I don't know if you guys have microscopes in the classroom or can obtain that, but go with more the algae, and see how algae are being eaten by, I don't know, tardigrades, right? That's like super awesome right there. And recognizing that that's being eaten by macros, and that's being eaten by bigger macros, and that's being eaten by bigger macros. I think that there's a food web is just fascinating, and it starts from plants. It starts from, and that's just the aquatic world, but you can also probably do it with the terrestrial world as well.
And Delita, it looks like there's one more question that came in. So how do you know when you're doing your research that the heavy metals are coming from the ospreys, or that you're getting from the ospreys are coming from Montana's food web and not where they've migrated from?
Great. Whoever asked that question, but careful because we might stay here till 6 p.m. with me. So what I didn't have the chance to tell you guys, and that's something that, of course, when in our curriculum we do talk about, is the life cycle of an osprey. The ospreys here in Montana are migratory. They're here only for the spring, summer, and fall. So they have this perfect setup where they're here for reproduction, and then early fall, female usually leaves first, says, okay, I've had enough, thank you very much, and she takes off to wherever that is. We've got some in Cuba and Mexico. The male then sticks around with the chicks for a little longer, and then takes off somewhere else. They take a break from each other, and which is really great. They're really excited when they meet here for life. They mate for life, and so they come back and they re-meet in Montana. We're not checking blood. We don't take blood from the adults because, right, who knows where the heavy metals came from. We're only checking the blood for the chicks. And so these chicks were fed the first fish, and the last fish before they fledge is on that nest. And that nest, for the most part, ospreys just, stay and fish within a mile radius from their nest. And that also makes it easy for us because the fledgling or the nestlings can't fly. And so we get up there with a bucket truck, we grab the chicks, there's nothing they can do about it, and we bring them down so that the audience can see the work that we're doing. Did I answer the question? Okay, perfect.
Okay, that was amazing. So much to talk about. We're going to go into breakout rooms for grade bands so that we can get a little deeper into this material. And our awesome guest speakers, you are welcome to stick around. You're also welcome to go get some dinner, fish within your mile radius, or whatever you need to do. So whatever works. And Michelle's going to send an invite to you for a breakout room. If you are in 5 through 8, we're going to stay here in this room.